someone sent me an email and asked me if I can say something about fluorescent protein, and I thought it would be interesting maybe to highlight a few important papers on fluorescent protein, and I answered yes before choosing the paper, and then I realized it was super hard to choose which paper I should highlight. So I decided to adopt another point of view. I would see if the pointer is working. Yes, it's working. And especially when I put in Web of Science green fluorescent protein or just fluorescent protein, I just realized that after 2000, the amount of paper was really huge. I knew that already, but it's really a lot of papers, and it's not so easy to identify which are the most important milestone. It's much more easier before, at the beginning of the 2000, because the number of paper were less. Here it's a log scale. Is the pointer working? Yes, here it's just a log scale. And if you try to identify the milestones, you can try to think about what could be done in the field of the discovery of the wild-type fluorescent protein. Most of the development based on fluorescent protein are due to the relationship between the structure and the photophysical properties. So it's also interesting to highlight the discovery of the first structures. And a lot of advanced versions of monomeric FPs have been developed, and those monomeric FPs are essential to develop functional imaging. And in those milestones, a very important milestone is missing, maybe the most important one. It's the founding of the GDR Mifobio 20 years ago. So it's an important milestone also for me, because that at that time, as a young assistant professor, I also entered in the field with a total different background. And all the networking, workshops, meeting organized by the GDR was really a great occasion for me to get in the field. And I hope that for the newcomer in the field, this Mifobio will be also a great occasion to see how broad can be the field, and I think it's a very good opportunity. So the next slide are quite conventional, and they have been already displayed by some of the previous speakers. So the story starts quite early in the 70s, even a bit earlier in the 60s with Shimomura. Shimomura identified where the luminescence of the jellyfish was coming from. And in fact, the wild-type AVGFP converts to green the bioluminescence of ichorine in the jellyfish photosite. So Shimomura got the Nobel Prize of Chemistry with Martin Chalfie, and the idea here, it was 30 years later already. The idea was to clone the gene. At that time, it was not as straightforward as now, to clone the gene of the AVGFP that has been done by pressure, and Chalfie had the very good idea to put the gene in a transparent organism. And that's the cover of Science, and you see that the GFP is folded and bright in a transparent C. elegans. So I think that's really the starting point of the story, because GFP the GFP fluorescence is genetically encoded. And in fact, most of the time, GFPs in general are compatible from live cells to organism. It's possible using the targeting sequence or a protein of interest to label specifically something in the cell. It could be an organelle, it could be a specific protein, but it's also possible to do genome editing and to develop, like here's a famous image of the fluorescent mice. So since the discovery of the AVGFP, a lot of fluorescent protein have been discovered, and in my first slide, I highlight the discovery of DSRAD, which was the first non-bioluminescent fluorescent protein discovered in a coral in Discosoma species, and since then a lot of fluorescent protein have been discovered in nature. And at the last Mifobio in 2021, we had the chance to hear Nathan Schenner, and he shared with us the discovery of other fluorescent other fluorescent protein coming from homologue of Ecoa Victoria. So all the fluorescent proteins discovered so far have a rather low sequence identity, but share a common structure, and it's a beta barrel crossed by an alpha helix, and this helix bears a chromophore, and the chromophore just results from the autocatalytic reaction, which lead to the cyclization of the residue and the chromophore. Two residues are conserved in position 96 and 222. They are very important for the maturation of the chromophore. And here I would like to stress that this chromophore, from a chemical point of view, as here a phenol, it can exist in two protonation states, phenol and phenolate. It's very important for the properties of the protein. But there is also here a double bond, and so there are two isomers, and cis and trans isomer. And those properties of the chromophore, which is embedded in a cavity, trigger all the properties of the fluorescent protein. And in order to match the performance of the fluorescent protein with application, the wild-type FP have been extensively engineered. That it was too hard for me to highlight and to pinpoint some article, because there is a strong interplay between the chromophore and the protein, and it's possible to introduce mutation either in the chromophore, in the protein, to tune the property, either the optical property or the chemical or the biochemical characteristics. So a lot of efforts have been made the last 25 years to get bright labels for multicolor labeling, to develop fluorescent protein as donor and acceptor adapted spectrally for FRET application, to have a specific environmental sensitivity for fluorescent protein, for tailored design of biosensor, but also to further expand to the red the spectral window for multi-labeling, but also for in vivo application. And last, it's very important to have FPs with controlled photoreaction for either super resolution or specific application. And a great actor in that field at the very beginning was Roger Chen, and he shared the Nobel Prize with Shimomura and Martin Chalfie, because he really expanded the palette of the fluorescent protein. And I can stress one of the examples we saw this morning with the cyan fluorescent protein. So at the beginning, in the first article in the late 90s, the ECFP was published, so it has a rather low quantum yield of 0.4 and a very short lifetime, which is also multi-exponential. And it took several rounds of engineering to lead to the M turquoise and to adapt the cavity of the chromophore to the bulky tryptophan here, which replace the phenol group of the tyrosine in the protein. So it has already been highlighted this morning, but there is a very powerful tool to choose a fluorescent protein, which is the FP base, and you can sort your protein you can sort the fluorescent protein by color, but also by epsilon, quantum yield, lifetime, and other parameters. And Roger Chen was also very active to develop smart labels and biosensor, and I think it was very important to start the field. So it's possible to build biosensor to exploit the intrinsic environmental sensitivity of FPs. They are sensitive to chloride. They might be sensitive to pH or reactive oxygen species. They have been engineered to get a specific sensitivity, especially for circularly permeated FPs, which we saw already several examples. And maybe one of the first examples in the early 2000 was with the first generation of G-CAMP. But it's also possible to design Foster resonance energy transfer based on two fluorescent proteins, and in that case, the FP is not responsive, it's just an optical reporter, which will transduce the structure and of the conformation of the proteic sensing model here, which is between the two fluorescent proteins. And one of the first examples is also for calcium sensing. It's called Chameleon. So in the next 25 years, the idea was to expand the function of those biosensors to render them highly specific to a signaling event. And there are biosensors now for almost everything in live cell. The idea is always to monitor several parameters in a single experiment. We already had the example in the previous previous presentation yesterday and also this morning. Ideally, it's important to be able to have the information in a whole organism. We had an example in the previous talk, and to have a rapid response. And of course, it will be ideal to be compatible with super resolution and other imaging strategy like optoacoustic imaging, for example. So we already saw some example of what we can do now. For example, Michael Lin yesterday showed us the latest version of the ALSAP voltage indicator. This morning, Joachim Goedart showed us the M-turquoise-based calcium sensor called FLITS. We will have a presentation from Julia Bertolin on Monday on thread-based biosensor for L3-3B and kinase. And Monday also, in the same session than Julia, we will have a presentation from André Steele. We will probably show us what can be done for super resolution and other imaging strategies. Okay, another field which is also important that has been developed based on fluorescent protein and based on their very impressive ability to be a photoactivated or photo switch. And I pick up only two examples, and those examples are also very old examples from the early 2000s. The first one was a discovery of the PA-GFP, so the photoactivable GFP. So with one mutation, it's possible to render a dark fluorescent protein bright. It involved a photochemistry step, so the decarboxylation of the glutamate 222 that I show you in the first slide. And this decarboxylation will stuck the fluorescent protein somehow in the anionic fluorescent state. Here you see here you have the absorption band of this state, and you see the fluorescence of the protein. So with a UV light, you can go from a dark state to a very bright green state. The other discovery, which was also very important, roughly in the same year, is a discovery of DROMPA. So DROMPA is able to switch between a dark state and a bright state. And in that case, it's a combination of protonation and isomerization of the chromophore. So here, the band here, which at 500, which is also absorbing, but which is also the absorption band, the fluorescent band corresponds to the anionic C state, and it can be switched off, and the chromophore is isomerized and protonated in the off state. And it's possible to turn on and off this fluorescent protein, and you can see here that there is some bleaching. So a lot of nanoscopies are based on the photochromism of fluorescent protein. I put some of them here, and that's why in 2014 the Nobel Prize of Chemistry was given to physicists, because their improvement was really based on the chemistry of the fluorescent protein. So FPs are plastic and robust to residue substitution. We already saw some example in the lecture today and yesterday, but they experience a very complex chemistry and they are rather big. And for the future, we can imagine that it would be great to have FPs with a high level of maturation to have a better photon budget. And it has been a bit discussed this morning, and it's not easy to screen. This parameter is not easy to screen. It would be great to have FP with efficient and controlled photoreaction. It's super important for the development of nanoscopy and new contrast. And it includes also the reversible photo bleaching, which is highly limiting the photon budget for, I think, all techniques. I think that the development of FP-based biosensor is not finished yet. And of course, it would be great to have fluorescent protein which are compatible with multimodal application. We saw, for example, this morning that the combination of super resolution and electron microscopy can be super powerful to resolve the structure of protein and protein complexes in cells. So if I focus a bit more on the graph, on the graph with the publication I showed you at the beginning, we see that, okay, the maximum of publication has been reached maybe in 2010, and now it's a bit decreasing. Now it's not in the log scale, so it can be seen more clearly. And it might be because some hybrid tag, which are a combination of genetically encoded parts and organic dyes, are emerging and are now widely used also. And we had some example in the presentation of Luke LAVIS today, just before this presentation. And I'm sure we'll have also other example on Sunday with the tool developed by Arnaud Gauthier with the FAST, the split fast, and the nano fast. So before I give the microphone to Luc for a presentation on organic dye, I just want to advertise, if you are interested in biosensor, there is a workshop organized, it's a workshop organized by Mifobio, by Imabio also, and it will be in June in Boulogne-sur-Mer, and you see the weather in Boulogne-sur-Mer is almost the same than here in Gien. Thank you very much.
All right. So I was asked just to give a brief overview of the last 20 years of small molecule fluorophores. And so what I thought I'd do is start with this slide that I made back in graduate school. So it's not quite 20 years ago, it's 15 years ago. But it sort of reflects the state of the art since this review article was looking backward. And in this chart, I show a lot of just general fluorophores, nothing too fancy. And that's sort of what was going on at the time. And so if you think about fluorescent dyes through the decades, it started, like I mentioned earlier, with these classic fluorophores in the 19th century and the early 20th century. And then the field got another boost in the 60s and 70s through laser dyes, where they took some of these classic fluorophores and started putting different substituents like fluorine or fused rings. And then in the 90s and early aughts, companies started basically taking the laser dyes and just adding sulfonate groups to make them more soluble so they'd behave better on antibodies. And so that was sort of the state of the art 20 years ago. And I just like to show this slide because, you know, there's sometimes if you look at these companies, they aren't really doing anything new. They're just trying to get around patents. So one example of this is just 488 excited fluorophores. So most of them are based on this rhodamine 110 that I mentioned earlier. This was in the original rhodamine patent from 1887. Alexofluor 48 is just sulfonated rhodamine 110. Dialite 488 is just Alexofluor 48 with this extra tail here, again, to get around a patent. Ado 48 is just Alexofluor 48, but with the linker here instead of down here. And then even newer dyes like CF 48 just combines this, and they just put the linker here with a little peg chain. Likewise, some of the other dyes, here's Ado 590, here's Alexofluor 594, basically this very similar structure. Here's Psi5, and here's Alexofluor 647. So also to think about something that happened almost 20 years ago, you know, it's as a dye chemist, sometimes I feel like I'm taking crazy pills because the dyes are all the same, it seems like. And in addition, back then we had classic labeling strategies. So sort of the original amine reactive labels were things like FITSI or TRITSI or Texas Red. And now these are not widely used, but they persist in our filter sets. And that's basically why we still use the words FITSI and TRITSI. And now we use more NHS esters, but that was done really in the 60s, or maleiamides for reacting with amines or thiols. And so what really changed in the last 20 years? So as I mentioned in my earlier talk, there was some improved chemistry. We also had new labeling strategies. And then I think a move to more complex biological systems. And because of that, the focus in the field, interestingly, has moved back to these small cell permeable dyes. And so one of the new labeling strategies was, of course, click chemistry, which won the Nobel Prize last year. And so there are a couple different flavors of this. Azide alkynes, either copper catalyzed or strain promoted. And you can buy really almost any fluorophore now with either an azide or an alkyne. And then a newer click chemistry approach are these tetraene transcyclooctane systems, which are really fast. Again, you can buy many fluorophores with these things on them. And the nice thing about this chemistry is, even a biologist can do this. You just mix two things together and they click. And you can use genetic code expansion now to install these sorts of motifs even into proteins and then click on dyes. And they're used for cell proliferation assays or purification in addition to labeling. Of course, another important thing that's happened are these hybrid small molecule protein labeling systems. And these come in two flavors: non-covalent, so things like FAST or the fluorogen activating proteins, and then the covalent labeling strategies, the Halo tag developed by Promega and the Snap tag developed by Kai Johnson. And so basically what's happened is now in the last 15, 20 years, we've moved to small, simple fluorophores without all this junk on it so they can get into cells and animals. So SIR, developed by Kai, and some of the dyes developed by my lab. All right, and so just thinking, what is this chart going to look like today or in 20 years? So I think in 20 years we'll be discussing how chemists finally figured out how dyes bleach and then stopped it. So understanding and preventing photobleaching, so we have super stable fluorophores. It's still shocking that we don't have a really good idea of how even fluorescein or rhodamines bleach. We're also going to push dyes into more and more complex systems. And so to do that, we're probably going to have to borrow principles from medicinal chemistry and then use those to make bioavailable dyes, just like we make bioavailable drugs. And then we'll also be talking about revisiting and improving cellular stains and other things. I think Andre does some beautiful work in that area, making dyes activatable with light or enzymes or different analytes. Meul does some beautiful work there. And then we need more hybrid small molecule protein-based systems. And Arnaud, of course, is doing awesome work there. All right, so with that, thank you very much. That's my take on the last 20 years of fluorophores. Happy to chat about this offline. Thanks.
I was wondering, what do you think of these self-healing fluorophores and all these kind of things?
Okay, I wasn't prepared for questions, but no. Yeah, so yeah, I didn't mention the self-healing fluorophores. Those are just dyes that typically have triplet state quenchers attached to them. I think, we haven't worked on, I think those are more useful for cyanine dyes and other fluorophores that are really susceptible to destruction by singlet oxygen. In our cases, sometimes we can get better results by installing deuterium or other things like that. I think there's still something to be learned, though, from those fluorophores, and we still don't understand how dyes bleach, how important is the triplet state, what's really going on. So it's going to take someone who really wants to understand photophysics and then try and fix whatever is going wrong. But I still think we don't have a good handle on how dyes bleach, and maybe the self-healing dyes will help us get there.